In this video, we present the accessories of the CYANElite 290 and the analyzer's first use. We have a separate video on the system configuration. On the right side, you find the hydraulic panel, which is completely segregated from the electronic components. Liquids are aspirated and dispensed by a convenient cartridge pump system. When you lift the plexiglass cover of the analyzer, you find a removable reagent tray on the left side and a sample tray on the right side on a cooling plate. The reaction segments are on a rotor under the fast access cover. At the back side, you have the on-off switch with the power inlet and the USB-B port to establish the connection between the analyzer and the computer. Ensure the power outlet has a stable tension. We always recommend to use an electronic stabilizer and a UPS to cope with any instability. All additional peripherals such as a keyboard, a mouse or external printer are connected to the computer. Verify if the CYANElite is installed in a suitable location. First, the surface must be stable, horizontal and vibration-free. Ensure you have at least 50 centimeters of open space around the device for air circulation. The surroundings should be dust-free and between 16 and 30 degrees. Avoid direct sunlight and keep the device out of the airflow. Ensure at least 3 meters from any electromagnetic radiation sources. And humidity must be between 10 and 80%. Non-compliance with these recommendations can have a negative effect on the performance of the analyzer. The onboard capacity of the reagent tray is 19 reagents and 1 diluent bottle. The reagent tray fits 3 types of reagent containers: the 35 milliliter bottles, the 15 milliliter bottles with a plastic adapter to fix it in position, and the 2 milliliter tubes which fit in a metallic adapter. The sample tray can fit 10 samples. It is compatible with 5 milliliter primary tubes with a maximum height of 75 millimeter and 1 milliliter cups placed in a specific metallic adapter. Using the adapters is crucial to avoid needle crashing due to shifting bottles and dropping of containers when transferring their trays to the fridge for storage. Switch on the analyzer and then the computer. Double-click on the high icon on the computer's desktop to launch the software. Press high again in the pop-up. At first launch or when connecting a different computer, a communication error might appear. In this case, select the available COM port from the drop-down menu and press Retry. The login screen will appear. Type the username user or administrator and insert the password. The reagent menu opens by default. You start the working day by verifying if the washing solution container is sufficiently full for your daily workload. The washing solution cleans the hydraulic tubes and removes contamination from the needle. To fill the washing solution bottle, first unscrew the green cap with hydraulic tube and level sensor and place it aside on a tissue. Add 5 milliliter of concentrated washing solution to 2 liters of distilled water to obtain a 1 in 400 dilution. We recommend doing this in 2 steps. Fill the container with 1 liter of distilled water. Add the 5 milliliters of concentrated washing solution. And then add the 2nd liter of distilled water. Screw the green cap back on the washing solution container. Also start the day with an empty waste container. Disposal of waste must be done in compliance with the local regulations. Then log both of these actions in the software. Navigate to the Maintenance menu and press the Replace Container button. Press Replace Containers again. Confirm the actions performed: refill of the washing solution container and/or replacing of the waste container. Remember, the liquid levels in both containers are in balance. If one container is empty, the other one will be full. Press OK to confirm. If the washing solution container has been refilled, the analyzer will now prime the hydraulic system. The status bar shows the progression of the priming. This task takes less than 5 minutes. Before running tests, it is necessary to load the reaction cuvette segments in the cuvette rotor. Press the cuvette replacement button on the bottom taskbar. Select each segment one by one and check if they are on board. Press the Replace button to indicate they are well placed. We recommend replacing the cuvettes indicated in red or blue. We have another video available on the cuvette replacement. It is important that the configuration on screen matches the reality. The reaction cuvettes in the CYANElite 290 are single use. Don't reuse the cuvettes. After use, the software highlights the cuvettes in red and will skip them from use while performing tests. The auto-zero procedure in the CYANElite is unique. The auto-zero is specific to a cuvette segment lot. That means that you must only auto-zero when switching lots. The lot is indicated on the cardboard box of the cuvettes. During this procedure called the cuvette realignment, the analyzer fills 24 cuvettes, which is one segment, with washing solution. Then aligns each individual cuvette in front of the optical beam and measures the optical density. The average optical density value is used as a reference in endpoint calculations. When necessary, press the cuvette realignment button. After 10 minutes, the auto-zero is completed. The starter kit provided with the CYANElite 290 contains all accessories used in this video. Thank you for your attention. Watch our next video too.